gamma-induced STAT1 activation as such. So having tested all these different inducer STAT combinations, we zeroed down to interleukin 6 mediated STAT3 activation, and we believe that the effect of node 1 is quite prominent with STAT3. But the question arises at that time that is this effect only prominent to STAT3, or it is also prominent to interleukin 6 as well? So to accomplish this, what we did is, under similar experimental conditions, we tested STAT3 activation by interleukin 10. And under similar experimental conditions, we see over here that the overexpression of node 1 results in no significant effect on interleukin 10 induced STAT3 activation as such. So at this point of time, we believe that node 1's suppressive effect is not only specific to STAT3, but it is also to interleukin 6. So, to supplement our data in HEC293 cells which were overexpression studies, we conducted silencing experiments in THP1 cells. And what we observe over here is that upon siRNA-mediated knockdown silencing and followed by interleukin 6 induction, we see a quite enhanced STAT3 activation. This definitely supplemented and boosted our overexpression studies in HEC293 cells, and we started believing that yes, these effects are quite genuine, and node 1 really leads to suppression of STAT3 activation as such. And under similar experimental conditions, we also tested the real nuclear translocation of STAT3, that how much of the activated STAT3 is there under similar experimental conditions. So these are, as like the previous slide, 72-hour knockdown of node 1 under comparative controls, followed by interleukin 6 time point inductions, and again at 15 minutes we see a quite enhanced STAT3 activation under these DNA binding assays. So altogether, these studies really made us believe that node 1 has a suppressive effect in the JAK-fascinating cascade, and in specifically interleukin 6 mediated STAT3 activation. So having said that, what we wanted to know that under these classical depicted picture of the NLR activation, these NLRs, as you would see, get activated when they recognizes these breakdown peptidogglycan compounds. So what we were interested at this point of time was to investigate that these suppressive effects that we observed on interleukin 6 mediated STAT3 activation by node 1 is to what extent it is affected by this known ligand of node 1. So in literature it is defined that node 1 recognizes gamma-D-glutamine, so diaminopedic acid. This is a peptidogglycan component mainly from gram-negative bacterium. And what we did is first make again overexpression studies in HEC293 cells, followed by STAT3 Western blots over here. And what we observe is that overexpression of node 1 results in a sustained attenuation of the phospho-STAT3 signals compared to anti-vector control. And under similar experimental conditions, when we use the ligand as such, upon analysis we observe is that the presence of the ligand makes no significant effect on this observed suppression of STAT3 activation as such. So in our studies, this receptor is acting independent of its ligand. So in a way, it is no longer acting as a receptor. Rather, probably, what we would say here is, in our setups, node 1 tends to probably have an adapter-like role as such. So to supplement these data and to make strengthen it further, we are interested in domain-specific studies of this so-called receptor. So going back to the structure over here, what we will observe that this molecule is divided into 3 specific domains as such. The leucine-rich repeat domain, which is known to be responsible for recognition of the specific ligand, though how this domain interacts with the ligand is not yet elucidated completely. This is followed by the central NBD domain, the nucleotide binding domain, which has an intrinsic ADPase activity responsible for the oligomerization and overactivation process, which is followed by the effector cascade recruitment domain via which this molecule interacts with its downstream partners in the cascade as such. So what we did is we thought that we would use different deletion constructs. So to start with, we used a construct not having the ligand recognition domain, which essentially make sure that there is no interaction with the ligand as such. And what we observed over here is that the suppressive effect on interleukin 6 mediated STAT3 activation is retained, as seen in the wild type case. This makes sense because in the last results, we observed that these suppressive effects are independent of the ligand as such. So when you are deleting the domain responsible for such a recognition and still having the suppressive effects retained, makes sense. Then what we did? We used the construct not having the effector domain, which essentially abolishes the possibilities of any downstream interactions with the supportive kinases and other molecules. And what we observe over here, the effect is still retained, as in the case of wild type or in the case of the construct not having the ligand recognition domain. Doubts raises that even after deleting the effective domain, the effect was still retained. But what do we observe over here? What do we exactly observe over here is that both these domains have the central nucleotide binding domain, which is responsible for polymerization and overactivation properties. But in this stage of dilemma, we decided to move ahead with further studies having individual domains. So what we initiated was just to have the construct having the ligand recognition domain alone, and we observe over here is that the effect is completely abolished. There is no suppressive effect on the STAT3 activation mediated by interleukin 6. What we did then, we used the construct having the only effector domain. So what we observe over here, again, the effect of suppression of interleukin 6 mediated STAT3 activation is again lost completely. And that do not answer our queries that how this suppression is actually mediated by this molecule. So what we did next